afternoon, students. This is the continuation of the genetics chapter, Mendel experiments on inheritance. The basic principles of genetics were discovered for the first time by Mendel in mid-19th century. He was an Austrian monk and conducted breeding experiment on garden pea. Mendel had selected garden pea for three reasons. First, many varieties were available in alternative form of characters. Second, varieties were available in pure form that breed true. Third, peas are normally self-pollinated, but self-pollination could be prevented by removing the reproductive parts of plants and can be cross-pollinated artificially. The Mendel has uh, cross-pollinated between the two contrasting characters, and he got the same result in different examples. Like few characters are given here: flower color purple white, seed color yellow green, seed shape round wrinkle. On all these contrasting characters, he did different practical work and he got the same result. So just one example I am giving here, that is, he has taken a axillary flower bearing pea plant, that is represented as A, and terminal flower bearing, that is represented as small a, and he crossed between these two plants. In F1 generation he got all seed with axillary flower bearing plant, and same seeds were shown in second generation, that is A, capital A and small a, that represent axillary flower bearing plant and terminal flower bearing plant. So in this, here you can see in this chart A and this A. This was crossed between this A and A. It became pure axillary bearing flower plant, and this A and A. So this is axillary flower bearing plant, but is the carrier of terminal plant also. And in the same way, with this A multiply this, we get this, and with this we get this. So this, the last one he got, that was the terminal bearing flower plant of P. So here you can see this is the pure axillary flower plant, and this is axillary but dominated with terminal, and this also the same, but it is the pure terminal bearing flower plant. So the ratio for phenotype and genotype is, for phenotype 3:1, means three plants were axillary flower bearing plant and one was terminal flower bearing plant. So in genotype ratio 1:2:1, means this is 1, these two are similar, so 2, and this is 1. 1:2:1. Always remember that in all cases, all like this experiment, he got the same result, that is for phenotype 3:1 and for genotype 1:2:1. Case 2, one more example we take here, that he took the pure tall TT pea plants crossed with pure dwarf plant TT. In the first generation, he got all tall plant, but all were the carrier of dwarf plant 2. In F2 generation, the cross between that tall plants which were the carrier of the dwarf plant, the same result were got. Phenotype ratio was 3 is to 1, genotype ratio 1 is to 2 is to 1. So he did number of experiment related to this and he got the same result. Next, dihybrid cross. For this he has taken the two pairs of contrasting character, round yellow crossed with wrinkled green. Round yellow, wrinkled green. In F1 generation, he got the result as all round yellow seeds. And in F2 generation, he took this crossed between these plants were taken. So the gametes for this experiment were prepared in this form: capital R, capital Y, capital R, small y, this capital R, small y, then capital Y and small r, then small r and small y. This is for female sex and same were taken for the male sex. And if you multiply with this, you get this. With this if you multiply, you get this. So same way with all this we multiply and get here this chart. And you can see the color of the seeds, how he got in different way. All you see the round yellow seeds were 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. And round green seeds were 1, 2, 3. And wrinkled yellow seeds were 1, 2, 3. And wrinkled green were found 1. So here the genotype is very complex, but phenotype ratio is 9 is to 3 is to 3 is to 1. So sometimes in the board exam they ask to write the phenotype ratio. So you have to write 9 is to 3 is to 9 is to 3 is to 3 is to 1. And sometimes like these questions will be given, you have to find out the progeny in this form. So please try. If not, when reopens this I will be doing once again. And next, Mendel's law of inheritance. There are 3 laws of Mendel and 3 also are asked to define sometimes in the board exam. One surely they will be asking out of 3. So please learn the definition of the Mendel's law. First law of Mendel is law of dominance. Law of dominance. This is the first law and how to define it. Out of a pair of contrasting character, one is able to express itself and other remain suppressed. That is known as law of dominance. Next is law of segregation. In that, two members of a pair of factors separate during the formation of gametes. Two members of a pair of factors separate during the formation of gametes. They do not mix, but they get segregated or separate into different gametes. The gametes combine together by the random fusion, that is for the zygote formation. This is known as law of segregation. And third is law of independent assortment. When there are two pairs of characters, the distribution of the allele of one character into the gamete is independent of the distribution of the allele of the other character. This is law of independent assortment and it is based on dihybrid cross. So please learn all these three definitions for the exam. Mendel application of Mendel's law. First law, a knowledge of basic Mendelian principles gives us an idea about the new combination in the progeny of hybrid and enable us to predict their frequency. Second, such information is of great importance to both plant and animal breeders for producing better breeds. And third, new types of plants with new combinations of useful characters can be produced by hybridization. Next is mutation. Mutation. Sometimes you will be asked to define mutation. Mutation is a sudden change in one or more gene or in this number or structure of the chromosome. That is mutation. Mutation alter the hereditary material of an organism's cells, result in change in certain characters or trait. For example, sickle cell anemia. It is a blood disease caused by a gene mutation. The mutation causes change in DNA resulting in the production of sickle cell anemia. RBCs become sickle-like. You may be knowing what is sickle. That is हसुआ जिससे घास काटते हैं धान काटा जाता है. That is sickle. So blood cells become sickle-like and it reduces the oxygen carrying capacity and the person get suffocated, enough pain in the body, etc. And the last is radioactive radiations also alter the gene structure and their effects can be seen generation after generation. An atomic explosion which had occurred during the World War II in 1945 in Japan, Hiroshima and Nagasaki, their effect is seen till today. Thank you dear student. Here I complete my lesson and you go through all the question answer of review questions and